Alright, hello everyone. As mentioned, I am going to be making a supplemental video for the remainder of Chapter 7 that we did not cover in class. Also, as mentioned, I do have the exam questions in hand, so I will tell you exactly what I want you to know for the exam for these particular topics that were not covered in class. Alright, so I left off here in class talking about motor unit recruitment, right? We know that a motor neuron and all of the fibers that it innervates is called a motor unit, right? And we can have multiple motor units for one muscle. It's called the motor neuron pool. And then we have this pattern of recruitment where we basically determine exactly how many motor units we need to recruit and able to produce a specific amount of force, right? If I want to pick up a weight, I want to contract my bicep, well, I need to recruit the exact amount of motor units required to produce the exact precise amount of force that I want to produce, right? The example I used in class was if I pick up a 5-pound dumbbell, I want to move my arm appropriate for that 5-pound dumbbell. I don't want to pick up that dumbbell as if I'm picking up a 50-pound dumbbell or else I'm going to smack myself in the face with it, right? We have to have that precise recruitment of muscle fibers based upon task demands, right? Depending on what it is we're doing, how heavy it is, you know what I mean? We need to have that precise control. And this precise control is determined at the spinal cord, okay? The decision to move a muscle can come from those higher brain centers, but in terms of that precise motor unit recruitment, that's going to be at the level of the spinal cord, okay? So moving along to where we left off, okay, we have two types of motor neurons. We have alpha motor neurons and gamma motor neurons. Everything that we've been talking about so far are alpha motor neurons. These are just the good old regular motor neurons that cause a muscle to contract, okay? Those are alpha motor neurons, okay? That's what we've been talking about this whole time. But we also have other types of motor neurons. Okay, so our alpha motor neurons, like I said, these are just regular good old motor neurons that cause contraction. They have large axon diameters and they innervate normal muscle fibers. Okay, they're called extrafusal fibers. These are just normal muscle fibers, right? So these muscle fibers that contract and produce force, those are called extrafusal fibers, right? So not only do we have two different types of motor neurons, we also have two different types of fibers, okay? Now these extrafusal fibers that are innervated by alpha motor neurons, again, those are just the regular old muscle fibers. They generate tension, they generate force by contracting, right? All of this stuff here is pretty much everything that we've already talked about. Now, gamma motor neurons, okay, these are going to be involved with some of our sensory information, okay, and I'll get into it in a little bit more in a couple slides, but just to give you the neuron kind of overview. So gamma motor neurons have small axon diameters, and they innervate what's called intrafusal fibers, okay, so these are another type of muscle fibers, right? The extrafusal fibers are just the regular muscle contraction fibers. Intrafusal fibers are part of this thing called our muscle spindle, okay? It's a sensory structure, okay? And we're going to talk about that in a second. So these little intrafusal fibers are part of the muscle spindle, which sends sensory information to the CNS, the central nervous system, about changes in muscle length, okay? So it tells us, where is our muscle? Is it lengthened? Is it shortened? What's going on with the muscle, right? It tells us that information. So these little intrafusal fibers, they do contract, okay, along with the extrafusal fibers, but they're not really generating any force, right? It's those extrafusal fibers, those thicker fibers, the just general muscle fibers that are doing the contraction, right? Those extrafusal fibers. These little thin intrafusal fibers, they also contract along with the rest of the muscle, but they're not really generating any force, right? They contract simultaneously with those regular extrafusal fibers, but they're too small. They're not really generating any significant tension or force or whatever, right? They're not involved with the process of the muscle contracting itself. They're kind of just contracting along the ride, right? When the extrafusal fibers contract, just the regular muscle contracts, these intrafusal fibers kind of contract along with them, even though they're not actually like producing any force or anything, right? They're a sensory part of the muscle, okay? And again, we'll get to the spindle in just a second. So we have something called proprioreceptors. Proprioreceptors are sensory receptors that basically tell our body or our central nervous system what's going on with our body position, right? Because even if we're not looking, if I put my arm out like this, I still know what my arm is doing, right? I can feel that my arm is stretched out. I can feel if my arm is half contracted or not, right? That's because of these proprioreceptors that are sensing this information and sending it and telling your body, right? They give us this information of what's going on with our body position, right? Otherwise, we wouldn't be able to move properly. We wouldn't have balance. We just wouldn't be able to move properly, right? We need to have a sense of what's going on in our body. So these proprioreceptors that tell us about body position are located in joints and muscles, okay? They tell us about muscle tension, so how much tension is on the muscle, how hard it's contracting, and also muscle length, okay? So the two proprioreceptors that we're going to discuss are muscle spindles and the Golgi tendon organ, okay? We talked a little bit about the muscle spindle here, but let's actually talk a little bit more about it. Alrighty, so the muscle spindle is located within the skeletal muscle connective tissue, and it responds to changes in muscle length, okay? Again, how stretched is the muscle? How contracted is the muscle? Just the position of the muscle. We have those intrafusal or thin fibers that we talked about that run parallel to just the regular normal muscle fibers, aka extrafusal fibers, right? Extrafusal fibers are just the normal muscle fibers, and then we have those intrafusal fibers that are just there along with it, right? So the intrafusal fibers run parallel to the normal muscle fibers, and they're innervated by gamma motor neurons. Okay, the gamma motor neurons stimulate the intrafusal fibers to contract simultaneously with extrafusal fibers, right? That's what we talked about here, right? The alpha motor neurons, they innervate the extrafusal fibers. These are our regular motor neurons, regular muscle fibers, just everything we've been talking about in this chapter, right? The alpha motor neuron innervates the muscle and tells it to contract. But these gamma motor neurons innervate those intrafusal fibers, the part of the muscle spindle, and tell the intrafusal fibers to contract, right? They contract alongside the extrafusal fibers, even though they're not really generating any force. They're just there for the ride, there to provide the sensory information. But because they still are a part of the muscle, they still have to contract when the muscle contracts. So these gamma motor neurons tell the muscle, these intrafusal fibers, part of the muscle spindle, to contract. Okay, so the gamma motor neurons stimulate intrafusal fibers to contract simultaneously with extrafusal fibers. So when the alpha motor neurons fire off and tell the muscle to contract, the gamma motor neurons are going to fire off at the same time, simultaneously telling the intrafusal fibers to contract, even though they're not actually producing any force, okay? And this is called alpha-gamma coactivation, right? Alpha and the gamma motor neurons activating together, okay? It's called alpha-gamma coactivation. Then with our sensory neurons, okay, we have primary nerve fibers that detect the velocity or speed of the muscle length changes, right? And then secondary nerve fibers detecting the length or the position. So the muscle spindle is not only telling us how fast the position, how fast the length of the muscle is changing, it's also telling us just the general position of the muscle as well. Okay, so that is the muscle spindle. But what is the purpose of the muscle spindle, right? We talked about how it kind of senses things and what it does, But what does it do? It initiates what's called the stretch reflex, okay? So basically, if a muscle is stretched too fast or stretched too much, this system of the muscle spindle is then going to tell the muscle to contract. So for example, if you have your arms out like this and somebody drops a big heavy weight into your arms, that's going to cause your muscles to stretch very fast, right? Your biceps, for example, other muscles too. But if you're holding your arms out like this, it's going to cause your muscles to stretch. And then your stretch reflex is going to kick in and be like, no, 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 we got to contract and resist that stretch, right? It's called the stretch reflex. So the stretch on the muscles causes a reflex contraction of the same muscle. And the way that we can demonstrate this reflex or this property is through the knee jerk reflex, okay, the patellar tendon stretch reflex. This is when you go to the doctor and they kind of like hit your knee with that little hammer and it causes your knee to kick out. Okay, that is the stretch reflex in action. Okay, so I'm just gonna go forward really quick to just show this picture, and then I'll go back and talk about exactly how it works. Okay, so here's the quadriceps, right? And when you go to the doctor, they're gonna hit on this little spot right here. Okay, right underneath the patellar tendon. Now this, you might be saying, well, what does hitting here do? Right? This is not stretching the muscle. Tapping this part of the knee does not stretch the muscle. So how is that triggering our entire leg to kick out, right? Well, so this tendon here is actually part of the quadriceps muscle, right? It goes over and this is actually part of the quadriceps, right? Now, because of where you're hitting, you normally only get a big stretch in this patellar tendon area if this quadriceps muscle is being like very, very stretched, okay? So because of the position of where you're tapping, it kind of like tricks the muscle into thinking that it's being stretched very dramatically, right? Because again, you're not really getting a very quick stretch on this tendon unless there's something big going on with the muscle, right? So the tapping at the doctor is like tricking the system. It thinks that by having this stretch right here, this quick stretch, that it means that the whole muscle is being stretched. So it basically triggers the muscle, it triggers the stretch reflex to contract the muscle, right? The tapping of the tendon in that spot is kind of tricking the muscle into thinking it's being very stretched, and therefore the stretch reflex kicks in and causes contraction of that same muscle, contraction of the quadriceps, causing your leg to kick out. Okay, that is how we test the stretch reflex there. So just going back, okay, how does this work? Muscle spindles are stretch receptors whose function is to correct for changes in muscle length when extrafusal muscle fibers are overly lengthened by excessive stretch. Extrafusal muscle fibers, again, just a fancy way of saying when the muscle is stretched, right? Corrects for changes in muscle length when the muscle is stretched. So when the muscle is stretched, the extra fibers are lengthened, the muscle itself is lengthened, and therefore the intrafusal fibers are also lengthened, right? We have the extrafusals and the intrafusal fibers. If the muscle gets stretched, it's going to stretch them all. When those intrafusal fibers are stretched, the increase in length of the intrafusal muscle fibers is detected and activates a sensory nerve fiber, right? There's actually a sensory neuron in there to detect. Otherwise, what else is going to detect it, right? If there's a stretch on the muscle, there needs to be something in there sensing that stretch and sending it somewhere so that we can do something about it. We can contract the muscle, right? So it activates the sensory nerve fibers, and then that sensory nerve information is going to go and activate both alpha and gamma motor neurons in the spinal cord. And what's going to happen if we activate alpha and gamma motor neurons? Well, we're going to have a contraction, right? The alpha and gamma motor neurons then signal both the extra and intrafusal fibers in that same muscle to contract and shorten to, again, resist that initial stretch, right? Therefore, when it contracts, we no longer have stretch on that muscle spindle, on that stretch receptor, which then we're fine, right? The muscle spindle isn't firing off anymore. The muscle is back where it needs to be. The stretch reflex did its job, right? This reflex results in contraction of the overstretched muscle to prevent damage from overstretching, okay? Now, this is also why when you are stretching intentionally, that you don't want to jerk your way into it, because you're going to activate your muscle spin. You're going to activate the stretch reflex, and it's going to resist that stretch. That's why you need to relax into a stretch. You don't want to go into it too quickly, or you're going to activate the stretch reflex, and your muscles are going to contract and resist that stretch. Okay, so this is why it's very important to be slow when you're intentionally trying to lengthen and stretch a muscle. So here it is. Here's how it goes. The slide just says structure and function. All right, so let's go step by step. First of all, what do we see here? Okay, here's a little up close picture. Okay, here's our extrafusal fibers, just the regular contractile muscle fibers that we think of when we think of a muscle. And then here's our intrafusal fibers, right? Those small intrafusal fibers. They don't look small here but this is a picture, right? We're trying to make it look large because we're examining it, right? But remember, those intrafusal fibers are smaller. They do contract, but they're not producing force, right? They're there for the sensory information. But here we have the muscle spindle, okay? So we have those gamma motor neurons, right, that are gonna be responsible for stimulating it to contract. But we also have those sensory neurons in there to detect the change in stretch, right? We have the gamma motor neurons that cause them to contract, but the whole point of the spindle is to be a sensory organ, right? You need to have that sensory information. So we need sensory neurons in there to detect the stretch and send it to the spinal cord so that we can cause contraction of the muscle. But anyways, so there's the muscle spindle, right? We've got some sensory neurons detecting what's going on, how much it's being stretched, how quickly it's being stretched, what the position of the muscle is, and then of course those gamma motor neurons that stimulate it to contract. These extrafusal fibers are innervated by alpha motor neurons, right? But again, remember, we have that alpha gamma coactivation, so they'll both fire off at the same time. But anyways, step 1, the muscle spindle detects stretch of the muscle, right? So, the muscle is stretching too far. We got to do something about that. The sensory neurons detect that stretch, right? It detects the stretch and conducts action potentials to the spinal cord. Why is it an action potential? Because action potentials are just the way that they communicate, right? The way that it's sending the sensory information is through action potentials. So the sensory neuron conducts action potentials. It sends that sensory information to the spinal cord. And now the sensory neuron synapses with the alpha motor neuron. Okay, so it's going to say, hey, alpha motor neuron, we got to contract this muscle to resist the stretch. So the sensory neuron synapses with the alpha motor neuron, and then the stimulated alpha motor neuron causes the muscle to contract and resist the stretch, right? And then you see here that there's also sensory information going to the brain. Why? Because they're also proprioreceptors. They're telling us the position of our muscles, right? We don't want to only know the position of our muscles to resist stretch. We just need to know it so we can live and know what our body's doing, right? So some of that sensory information goes to the brain just for general information, and then it also synapses on the alpha motor neuron to do its job with the stretch reflex, okay? Now, I just got finished showing how the muscle spindle will send sensory information and then synapse with the alpha motor neuron to contract that same muscle, right? If the quadriceps here are being stretched too much, then we're going to stimulate it via the alpha motor neuron to contract, to relieve that stretch. However, it's actually also causing inhibition of the antagonistic muscles. Antagonistic means the opposite muscles, right? So if we have contraction of the quadriceps, right? If we're stimulating the quadriceps to contract, then we're going to inhibit the antagonistic muscles. What are the antagonistic muscles of the quadriceps? The opposite movers, the hamstrings, right? If you stretch this is not a quadricep, if you flex your quadriceps and lift your leg, then you need where did I go? All right, well, I apologize for being rudely interrupted. It was a Zoom update that decided to update in the middle of using Zoom. So, seems pretty efficient to me. But anyways, where were we? So we were talking about how the muscle spindle is basically, its job is to sense stretch and then cause contraction of the muscle to resist the stretch. But we're also inhibiting the antagonistic muscles, right? So when we stretch that quadricep and therefore activate the stretch reflex and contract the quadricep, we're also causing relaxation of the hamstring, of the antagonistic muscles, okay? So here we have that, right? Here's the stuff that we already talked about, right? The sensory information, we sense the stretch of the muscle, right? That sensory information is sent to the spinal cord and it triggers the alpha motor neuron to contract the muscle and also the synergistic muscles as well, right? So not just that one quadriceps, but all of the quadricep muscles together. And then we are also inhibiting the antagonistic muscles, the hamstrings, to allow the quadriceps to properly contract, right? They won't be able to properly contract if the hamstrings are activated, right? So we see here we have this inhibitory interneuron. Okay, so this is how we're getting inhibition of the antagonistic muscles. So this inhibitory interneuron, right? The interneuron is just like a neuron in between some other neurons. Its job is to inhibit, right? When we activate this inhibitory interneuron, it's going to do its job of inhibition and inhibit those antagonistic muscles, right? Those opposite muscles. But it's done through this interneuron process, okay? So the sensory neuron stimulates an inhibitory interneuron. So we're stimulating or turning on this inhibitory interneuron. So now this inhibitory interneuron is going to do its job of inhibition and inhibit the antagonistic muscles, okay? But it's done through this inhibitory interneuron. So this is a little confusing here when we talk about stimulating an inhibitor or inhibiting an inhibitor, right? If this was if we were inhibiting this inhibitory interneuron, right? If we were turning it on, then it wouldn't be able to do its job of inhibiting, and we would not have relaxation of the antagonistic muscles, right? So just it gets kind of confusing, the wording, when you start talking about the inhibition of an inhibitor, excitation of an inhibitor, or whatever. You just have to know what you're looking at, okay? So just be careful with that terminology here, okay? We're stimulating something that's going to inhibit, right? So if we're stimulating this inhibitory interneuron, it's going to inhibit, okay? Now this is called reciprocal inhibition, okay? When we simultaneously cause the synergistic muscles to contract and resist the stretch, right, of the homonymous muscle. Homonymous just means like the muscle that we're talking about, right? The homonymous muscle receives the stretch reflex kicks in and causes contraction via that alpha motor neuron, right? The alpha motor neuron, and also the gamma neuron. It's just not showing here, right? So we get a full contraction, but we're also inhibiting that reciprocal inhibition of the antagonistic muscles, right? So if we're triggering the quadriceps to contract and resist stretch, we're also inhibiting the hamstrings, okay? And that is called reciprocal inhibition. Okay, now we also have what's called the Golgi tendon organ, or the GTO. The GTO is located within the tendons, and they monitor tension within the muscle, so how hard the muscle is contracting. This prevents muscle damage during excessive force generation, right? So this prevents us from generating so much force that we damage our muscles, that we rip them off, right? If we didn't have something to slow that down, then theoretically we could generate so much force that we damage our own bodies, okay? So the GTO prevents that from happening. It says, hey, this is way too heavy for us. We're going to inhibit this muscle. Let's relax this muscle so we don't hurt ourselves, okay? It makes you drop a heavy weight. So if you've ever experienced a very jerky movement while lifting a heavy weight, that's your GTO. That's your GTO kicking in and saying, hey, I don't know, this might be a little too heavy for us. I think we need to, stop contracting this muscle. Okay, so when we stimulate the GTO, it results in the reflex relaxation of muscles. Okay, so what we see is inhibitory interneurons send IPSPs, inhibitory postsynaptic potentials, to muscle fibers. This is just a fancy way of saying inhibiting muscle fibers, right? Because what's an IPSP? Right? An IPSP, an inhibitory postsynaptic potential, is pushing the cell farther away from threshold. And with a muscle cell, we're only going to get a muscle contraction, right? An action potential and a muscle contraction unless we hit threshold. So if we send inhibitory neurons sending IPSPs, we're pushing it farther away from threshold. We're causing that muscle to be relaxed, right? Excitation would be contraction. Inhibition is relaxation of the muscle. Okay, but again, that's just how it works. The same thing is happening here, right? Instead of sending inhibitory postsynaptic potentials, we're just sending excitatory ones, right? I don't know why we decided to start throwing in this terminology only for the GTO, but this is really how any inhibition is working, right? Any inhibition is working via IPSPs. Any excitation is working via EPSPs, right? But anyways, that's kind of getting off topic there. But anyways, so what's interesting though is with resistance training, we can actually oppose the GTO. Okay, so with resistance training, we kind of desensitize the GTO. So it's not so like on alert, right? If we lift something really heavy, our GTO goes, whoa, 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 this is too heavy. We need to relax this muscle, put that down. But if you resistance train, it becomes less sensitized. It goes, you know what? I think we could lift some extra weight. I think we're okay. It becomes a little less sensitive, right? This is a neural adaptation that allows for increased force production prior to GTO activation, right? So you can lift heavier weights before your GTO kicks in and says, hey, I think we should slow this down, right? And this is a neural adaptation because it's happening through neurons and sensory neurons and whatnot. So how does it actually work? Okay, the Golgi tendon organ, they are stretch receptors, right? They're sensing the tension on the muscle, whose function is to alleviate excess tension on contracting muscle. The GTO is much less sensitive to stretch and tension than muscle spindles and requires a greater stretch tension to be activated, right? Because the GTO is, the muscle spindle is like, hey, we're stretched, let's contract, right? It's active all the time. But the GTO is kind of like that last minute protective resort, right? We don't really need the GTO to kick in unless we're really about to hurt ourselves. So the GTO is less sensitive than the muscle spindle, and it takes more to actually activate the GTO. So with the GTO, what are the steps? When the muscle fiber contracts under excessive tension, right? We're talking about something where we might hurt ourselves. There's too much tension here. So the extrafusal fibers shorten. Literally, that is just the muscle shortens, right? The extrafusal fibers are just regular muscle fibers. when the muscle fibers shorten, the extrafusal muscle fibers shorten, it activates the Golgi tendon organs. When activated, the GTO sends information, impulses, action potentials to the spinal cord via sensory neurons. The sensory neurons that are bringing that contracting information synapse on inhibitory interneurons in the spinal cord. When we activate those inhibitory interneurons, we're going to inhibit that alpha motor neuron, right, which is the one that's responsible for causing contraction of the muscle, therefore producing relaxation of that muscle, right? When we inhibit the alpha motor neuron, the muscle is going to be relaxed, right? The alpha motor neuron is what causes muscle contraction. Therefore, this results in the relaxation of the contracting muscle to prevent damage from excessive tension, right? So this is essentially the exact opposite of the muscle spindle, right? The muscle spindle detected stretch and then contracted as a result. The Golgi tendon organ detected the stretch, but like the tension of the stretch, if it's too much, and then it causes relaxation, right? It's the opposite. So let's go through it. So here's our GTO, right? It's in the tendon, the Golgi tendon organ. And there's that sensory information, right? It's in there, it's sensing that tension, and then it's going to send that information to the spinal cord. Okay, so step 1. The Golgi tendon organ detects tension applied to the tendon, right? There's too much tension here. I think this muscle is going to we're going to get damage. I think we need to relax, right? The GTO is sending all that sensory information. The sensory neurons conduct action potentials to the spinal cord, right? That's just this fancy way of saying it's sending the information. Neurons send information via action potentials. The sensory neurons then synapse with inhibitory interneurons that synapse with the alpha motor neurons. So if we're stimulating this inhibitory interneuron, well, then we're telling it to do its job, which is inhibition, right? That's the job of the inhibitory interneuron. So we stimulate this inhibitory interneuron, which inhibits the alpha motor neuron. Inhibition of the alpha motor neuron causes muscle relaxation, therefore relieving the tension applied to the tendon, right? So if you think about it, this and the muscle spindle is kind of like a negative feedback mechanism, right? Too much tension, okay, let's get rid of that tension and relax the muscle. All right, so just like the muscle spindle, we have something else going on at the same time, okay? And it's called reciprocal excitation. So simultaneously, information is sent from the spinal cord to cause relaxation of the synergistic muscles, which is everything we just talked about, right? When the GTO is activated, it says, whoa, there's too much tension here. Let's relax this muscle so we don't hurt ourselves. So it causes relaxation of synergistic muscles and contraction of antagonistic muscles, right? This is the exact opposite. So if, for example, we're doing a bicep curl and there's too much tension, we're gonna hurt ourselves, right? Our GTO is gonna kick in and it's gonna say, hey, relax that, okay? Relax that bicep. But in addition to that, it's going to tell our antagonistic muscles, our triceps, to contract. Why? To assist in that movement, right? We can relax the bicep, but if we contract the tricep, we're going to have a much better relaxation of the bicep, right? So it not only causes the relaxation of that same muscle by inhibiting the alpha motor neuron and inhibiting the synergistic muscles, it also stimulates contraction of the antagonistic muscles, and it does that through an excitatory interneuron, right? So here we have inhibitory interneurons, right? So we're stimulating these inhibitors, we're getting inhibition, and then here we're stimulating an excitatory neuron. So This stimulates this, which stimulates the antagonistic muscles, okay? So again, it's like the exact opposite of the muscle spindle. The muscle spindle detects stretch and causes contraction of that same muscle and relaxation of the antagonists. That's called reciprocal inhibition, where the GTO causes relaxation of the same muscle and synergistic muscles and contraction of the antagonistic muscles. Okay? So what do I want you to know about these topics? Right? I'm looking at the pictures, or the questions here. I want you to know what the term alpha-gamma coactivation is. Where is it? Okay, that's when the alpha and gamma neurons contract together. I want you to know that that is part of the muscle spindle. Okay, that's specifically what I want you to know. I want you to know what the muscle spindle and the GTO do. Okay, so the muscle spindle initiates the stretch reflex. Let's see. Know the function of the muscle spindle and the GTO. The function is to prevent tissue damage. I want you to know that the GTO can be desensitized with resistance training. I want you to know that both the muscle spindles and GTO are both proprioceptors and therefore sensory receptors. Okay? And then I want you to know basically what each of these do. Okay? I want you to know which one does reciprocal inhibition and which one does reciprocal excitation. I want you to know where each one is located. Okay, so the muscle spindle is in the connective tissue, and the GTO is in the tendon. Okay, so I want you to know where each one is. I want you to know that they are both stretch receptors. Okay, they are both stretch receptors. I want you to know which one, what they each do. So which one causes muscle relaxation of the main muscle, obviously. Which one causes relaxation and which one causes contraction. I want you to know that they both contain sensory neurons. Okay. All right, moving along to the withdrawal reflex. Okay, I'll just put myself over here. Now, the withdrawal reflex, okay, this is a reflex that occurs in response to a painful stimuli. Okay, so if you put your hand on a hot stove, you have a reflex that's going to tell your hand to withdraw away from that painful hot stove. Okay, and by the way, the GTO and the muscle spindle, these are reflexes, right? The stretch reflex, the GTO, the withdrawal reflex, these are all happening in the spinal cord, okay? We don't really need the higher brain centers for these reflexes to happen. And there it is. There's the bullet point that exactly says that. So the way that the stretch receptor works, okay, so here's a little picture. Here's the painful stimulus. It's showing touching a pin, right? So this would be receptors in our skin. Sensory nerves send impulses that say, hey, this is really painful. They send impulses to the spinal cord. The interneurons activate the motor neurons to control the movement of muscle, to take that hand away from the hot stove or the pin. Okay? Again, here's that interneuron. Okay? So you don't really need to know what everything up here is called or whatever, but I do want you to know that these interneurons are playing a role, right? We have interneurons here with the GTO. We have interneurons here with the muscle spindle, right? They're just arranged a little bit different in different types of interneurons. Okay. And the withdrawal reflex also has reciprocal inhibition. So it sends excitation impulses to muscles that are involved with withdrawing away from the stimulus, and it sends inhibitory impulses to the antagonistic muscles, right? So if I put my arm down on a hot stove, it's going to excite the muscles responsible for pulling away from that stimulus, and it's going to inhibit the muscles that were used to put the hand on the hot stove, right? It's reciprocal inhibition. We're exciting some and inhibiting others. Now, we also have the crossed extensor reflex, which is part of this withdrawal reflex. So what we're going to see with this is the opposite limb supports the body during the withdrawal of the injured limb, okay? So for example, if you, what we're going to see on the next page, basically, if you put your hand on a hot stove, when this hand pulls away, it's going to cause the other hand or other arm to go in the opposite direction to essentially help you keep balance, right? If you were walking and you stepped on a tack or something, and you only flew back away from the tack, you're going to fall down and injure yourself. So this crossed extensor reflex kind of, it like helps your body keep balance. So even though you have the one limb withdrawing away from the pain, the other limb that doesn't have any pain or anything also has a response to keep you in balance, okay? The opposite limb supports the body, works simultaneously with the withdrawal reflex, okay? So this crossed extensor reflex is kind of part of this withdrawal reflex. So let's go through how that one works. Okay, so the crossed extensor reflex, a reflex that occurs in conjunction with the withdrawal reflex in response to a painful stimulus. It also encompasses reciprocal inhibition and reciprocal excitation, right? We talked about that. When your left hand touches a pin, sensory neurons are activated that synapse with multiple interneurons in the spinal cord that innervate and activate alpha motor neurons. It's kind of what we talked about so far with the other ones, right? With the GTO and the spindle. Flexor muscles contract and extensor muscles relax. Flexor muscles contracts and extensor muscles relax on the ipsilateral limb, aka the same limb as the painful stimulus. The contralateral limb would be the opposite one. Okay, so ipsilateral is the one that's experiencing the painful stimulus. Okay. Simultaneously, flexor muscles relax and extensor muscles contract on the contralateral limb, the opposite limb. Okay, so while this hand is on the hot stove and it goes away, it withdraws, right? It contracts these extensor muscles. The other hand is going to contract the other muscles, right? So it's like the exact opposite thing going on with the other arm, again, to maintain balance, okay? It causes the flexor muscles to relax and extensor muscles to contract on the contralateral limb, the opposite limb, thus the name, the crossed extensor reflex. This reflex produces flexion of the ipsilateral limb and simultaneous extension of the contralateral limb to help the body maintain balance during the withdrawal reflex. Okay, so here we have that here. Here's the painful stimulus, right? So this would be our ipsilateral limb. So we touch the pin, the sensory fibers tell the spinal cord to flex this arm away from the pin and inhibit the extensor, right? Because it was this tricep that caused it to touch it, or however it worked, right? But on the other side, we're going to see the exact opposite movement, right? In the contralateral limb, instead of stimulating flexion, we're going to inhibit flexion and stimulate extension, right? To go the opposite way to maintain balance. So that is the crossed extensor reflex. Here are a summary of the reflexes, okay? You can read through this. You don't need me to necessarily read through all of this to you, but one thing that you can note is the number of synapses, right? There's a lot of interneurons at play. So that's kind of what this is talking about. The stretch reflex, where's the stretch reflex? There was no interneuron, right? That sensory neuron went and synapsed right on that alpha motor neuron to get that response. But with the other ones, and the reciprocal inhibition part, we have an interneuron. Or the GTO, we have interneurons, right? Here we have interneurons, and so on. Okay, so that's what this means here. Now, what do I want you to know about these things? I want you to know that the withdrawal reflex occurs in response to painful stimuli, that it involves reciprocal inhibition, and that it works simultaneously with the crossed extensor reflex, right? It's part of that. I also want you to know this, which ones are polysynaptic and which ones are monosynaptic. Okay, and let's move on to chemoreceptors. Now the rest of this I am going to try and make very quick, okay? Muscle chemoreceptors, okay? So we just talked about proprioceptors that are telling us about the position and the stretch and the tension of our muscles and where our body is in space, right? That's what proprioceptors tell us. Chemoreceptors are telling us about changes in the chemical environment, okay? So muscle chemoreceptors are telling us about changes in the environment surrounding the skeletal muscle. So they're going to react to things like hydrogen, CO2, potassium, okay? So increases in any of these substances are going to stimulate the chemoreceptors. Now, why are these particular substances important? Why are they sensing these substances? Well, for one, when we are undergoing exercise, for example, or when we're very metabolically active, we're going to be producing CO2, right? When we're aerobically producing ATP, we're using oxygen and we're producing CO2. Or if we're doing very high intensity exercise and we're generating lactic acid and our bicarbonate buffer system kicks in, we're generating CO2. Hydrogen, right? If you're exercising pretty hard and you're generating lactic acid, you're going to get an accumulation of those hydrogen ions. And potassium. Okay, so these two are pretty straightforward, but this one is a little, it relates to what we talked about in this chapter. So if you recall, when we get action potentials, and action potentials are what lead to muscle contraction. When we're having a lot of muscle contractions, such as with exercise, well, we have a lot of sodium flowing in and potassium flowing out, right? So if we're just having repeated muscle contractions over and over and over and over again, we're going to start to accumulate potassium on the outside of the cell, right? We have that sodium-potassium pump that kicks in and puts everything back into place, but when we're doing something like heavy exercise, sometimes that sodium-potassium pump just can't fully keep up. So we start to accumulate potassium on the outside of the cell, okay? So that is why chemoreceptors respond to potassium as well. So basically, the increase in any of these substances indicate metabolically active tissues, okay, actively contracting muscles. So these chemoreceptors tell the central nervous system what's going on about the metabolic activity, right? Hey, these metabolites are building up, so I think we need to activate the cardiovascular and the pulmonary systems, right? So these chemoreceptors are very important for the cardiovascular and pulmonary response to exercise. As you may know or may learn, when our initial increase in breathing during exercise is not because we need more oxygen, it's because we need to get rid of CO2. So these chemoreceptors are very important in regulating the cardiovascular and pulmonary response to exercise, okay? The cardiovascular and pulmonary systems rely on this information so they can know what they need to do, right? Is there too much CO2 accumulating? Do we need to up the respiratory system? Do we need to divert blood flow to these muscles because they're active, right? It tells us information. So what do I want you to know about the muscle chemoreceptors? They detect metabolically active tissues. They assist in the pulmonary and cardiovascular response to exercise. And then I want you to know these three things they are sensitive to. And that's it for that. Okay. Last part, I'm literally going to breeze right through this. Okay, I'm really not going to talk about this too much. These are things that will be covered in neurophys a little bit more. But anyways, so now we're talking about what's going on in the brain with movement, right? We've talked a lot about these reflexes in the spinal cord, right? Everything that we talked about here is happening in the spinal cord, right? It doesn't require upper brain. Even motor unit recruitment, whoa, I scrolled way back. Even the motor unit recruitment is happening at the spinal cord, okay? So now we're going to talk about what's going on above all of that. What's kind of driving all of this. All right, motor function control. So the subcortical, which is just below the cortex, and cortical motivation areas, this is where you sort of get that initial drive to move. Okay, so on the next slide here, everything that's right here is basically everything I'm saying here, just in like a visual chart. Okay, so the initial drive to move, the initial decision to move, comes from this subcortical and cortical areas. Then the cerebellum and basal ganglia, they cooperate together to form a precise movement plan. Okay, the cerebellum is involved with fast movements and the basal ganglia involved with slow, deliberate movements. Okay, so we get that movement rough draft, okay, coming from the association cortex, the basal ganglia, and the cerebellum. Then the move message is sent through the thalamus to the motor cortex, then down the spinal cord for spinal tuning of the muscular response. When I say spinal tuning, I'm talking about that delicate motor unit recruitment, right? Remember in class, I talked about when we need to precisely recruit the specific amount of motor units to produce the specific amount of force that we want. Right? All of that is going on at the spinal cord. Those initial decisions to move are happening in these higher brain centers. But that actual fine-tuning of the motor neurons, right? The actual motor unit recruitment, where we decide how many we need to recruit to produce that force, that is happening in the spinal cord. Okay? So here we have that, okay? It gets relayed. We get this final executor of the motor plan, okay? Feedback from muscle receptors and proprioceptors allowed for fine-tuning of the motor program, aka precise movement, okay? So we relay through the thalamus, then to the motor cortex, which develops that final execution of the motor plan, and then the motor unit themselves, right, the alpha motor neuron and the fibers that it innervates, then does the actual execution, right? So you start with these higher brain centers, and then you end up with movement at the spinal cord, right? So here is spinal cord, right? This is where we deal with the actual motor units and the pattern of recruitment and the actual movement, right? The motor neurons, those alpha motor neurons, come off the spinal cord, right? And the recruitment pattern comes from the spinal cord. But again, that initial drive to move comes from the higher brain centers. So what do I want you to know about this? I want you to know these steps in order, okay? So know these steps in order. And that is it. Now we're going to talk about a couple more things and hopefully be done soon. Okay. So we talked a little bit about muscle fatigue, right? We talked about muscle fatigue when we talked a little bit about lactic acid, okay? Now central fatigue is not the same thing. So we have central fatigue and we also have regular muscle fatigue. So central fatigue is neural fatigue. It's not the same thing as muscle fatigue. It affects higher brain centers and or motor neurons. So basically, when we have regular muscle fatigue, if we're low on ATP or we have too much lactate accumulating and we're getting acidic and the muscle is not really contracting anymore. Neural fatigue is like when your nervous system gets tired or overworked, okay? And the way that happens is when we deplete our excitatory neurotransmitters in the motor cortex, resulting in reduced motor output, right? Reduced neural drive to the muscle. If we're not able to stimulate the muscle with those motor neurons as effectively, we're going to have a decrease in force production, right? That is central fatigue. It's not happening in the muscle. It's those brain centers that are like, whoa, we've been exercising too much. We're overtraining. This is too much. We're depleting our neurotransmitters and we're not able to fire off to those muscles as effectively. The central governor theory says that the central control system or the central center or the central nervous system regulates exercise performance, right? It reduces motor output to the exercising muscle and protects against catastrophic disruptions of homeostasis. So this is saying that we have this governor system in place to prevent us from excessively exercising and killing ourselves, right? If we just exercise way too much, we can't sustain that forever, right? So there's this theory that our central nervous system has a role in that to kind of slow things down. Research suggests that fatigue is...
Sorry, I didn't understand.
I didn't ask your opinion. Research suggests that fatigue is related to both central and peripheral factors. Okay, so basically it's saying that overall muscle fatigue is due to not only regular muscle fatigue, which we're going to discuss more in chapter 8, but we did talk about a little, right? Depletion of ATP or lactic acid and things like that, and central fatigue. Okay, so they both lead to the same thing. Both central fatigue and muscle fatigue lead to a decrease in muscle performance. It's just different ways, right? Muscle fatigue is things going on within the muscle. Central fatigue is the nervous system kind of tapping out. So what do I want you to know about that? I want you to know that it can be caused by overtraining. I want you to know that it decreases motor drive to skeletal muscle. I want you to know that it is caused by the depletion of excitatory neurotransmitters. And I want you to know that it is not the same. It is not the same as muscle fatigue. Okay? It's not the same. Central fatigue is not the same. All right. Let's finish this up here. The last few slides here, I'm literally just going to kind of read to you because if you see here, details in neurophysiology, you're going to talk about it in another class. So this is not even my place to go over. All right. The vestibular apparatus located in the inner ear. This is responsible for maintaining general equilibrium and balance, right? So our inner ear detects our head position, right? If our inner ear gets all wacky and mixed up, that's how we end up with vertigo, right? Because then it can't tell where you are in your position and it gets all freaked out, right? Your vestibular apparatus tells us exactly where we are in position. It maintains head position. It's sensitive to changes in linear and angular acceleration, aka head movement. And it also controls head and eye movement during exercise. The cerebrum. Okay, we have the cerebral cortex and the motor cortex. The cerebral cortex is where we store our learning experiences, the reception of sensory information, and the organization of complex movement. The motor cortex deals with our voluntary movement. The motor cortex controls motor activity with the aid of input from subcortical areas, such as the brainstem and spinal cord. Okay. The cerebellum coordinates and monitors complex movements. Okay, it incorporates feedback from proprioceptors. Okay, so our proprioceptors not only are doing their reflex things, but they're also telling our brain just where we are in space and whatnot, right? Those proprioceptors are not only good for reflexes we want to consciously know where our limbs are and what they're doing. Okay, so it has connections to the motor cortex, the brainstem, and the spinal cord. It may also initiate fast ballistic movements. The brainstem. Okay, we have the medulla, the pons, the midbrain, the reticular formation. Okay, this is something you'll go over in neurophys. It's responsible for many functions. Okay, your brainstem is very important. Okay, it controls your cardiorespiratory control and complex reflexes, right? So if you have a brainstem injury, you could die, okay? Sports-related concussions, okay? I'm not going to go too far into this. Basically, when you get a concussion, you get hit in the head really hard. You get a brain injury because of that traumatic force. Okay, it typically resolves in a few days. It may or may not involve a loss of consciousness, but it has a lot of negative effects. Okay, you could have permanent brain damage or death due to the swelling. If you get another concussion before the first concussion has healed, that is very bad. That could literally lead to death. I know if you're a football fan, you may have seen this happen recently, not really recently, but yeah, we see this happen in football too. Okay, same season, repeat concussion. And then there's late life consequences of repeated concussions. It can lead to Alzheimer's, other cognitive and mood disorders. If you've ever heard of CTE, right, we see that a lot with football, things like that, when you get repeated head injuries. Yeah, not good, okay? Here are some of the signs and symptoms of a concussion. Okay, you see that there's physical, cognitive, emotional, all sorts of effects. And then exercise enhances brain health. Okay, exercise enhances everything, right? This is kind of the common theme here. Exercise is good, just not too much exercise or, whatever. So exercise improves brain function and reduces the risk of cognitive impairment associated with aging. Regular exercise can protect the brain against Alzheimer's disease, other diseases, types of stroke. And how does exercise do that? It enhances learning and memory. It stimulates neurogenesis, which is the formation of new neurons. It improves brain vascular control and blood flow, right? More blood flow to the brain is usually a good thing. It attenuates the mechanisms responsible for feeling depression, and it reduces peripheral factors implicated in cognitive decline. So things like inflammation, high blood pressure, insulin resistance, right? These are things we typically think of as just kind of in the body, but they all have an effect on the brain as well. Okay, exercise also increases the production of something called BDNF, brain-derived neurotrophic factor, that is involved with the process of neurogenesis and learning and memory and all of that. So yeah, regular exercise increases brain growth factors, BDNF being one of them, and then we get a nice increase in brain health, which increase in brain health leads to enhanced overall health. Now, what do I want you to know about the last few slides? Okay, I want you to know what each of these brain centers do, right? So literally you can just like print out these slides and, so which one maintains balance and equilibrium, which one is the origin of voluntary movement, which one coordinates complex movement, and which one has the cardiovascular and respiratory control centers. I also want you to know the potential implications of concussions. Okay, so we can have swelling, we can have death, we can have diseases, mood disorders. And then I want you to know the potential benefits of regular exercise on brain health. Okay, so I want you to know basically these bullet points right here. So that is it for the supplemental video. Again, I'm sorry if it's a little bit long. Like I said, to make it worthwhile, I'm telling you exactly what I want you to know about those things that we cover in the video. Please let me know if you have any questions or if you aren't understanding something that I'm explaining in the video or whatever it is. So yeah, just reach out if you have any questions. I'll be more than happy to help, and I hope you have a wonderful day.